Okay, hi everyone. I hope you are having a great day. And so I want to get back on where we left off for reviewing the MCAT material. So I find myself uh, being overwhelmed kind of by these chapters and the fact that there are so many flashcards. For example, we finished the biology chapters. However, there's like 850 or so flashcards just to review, and like 400 of them were like duplicates of the ones that we did before. So I'm and also I'm kind of slacking on the doing the flashcards. it kind of gets it can get boring pretty fast. So now I want to kind of switch up things just to get back on the fundamentals and be able to finish all these chapters and concepts and test ourselves constantly. So now I'm running, by the way, I'm running on zero sleep right now. And if I get sleepy, I will just take a quick nap or a couple of hours of siesta, and then we can continue. But the plan is to just study these concepts in however possible, either through flashcards, through the overview that we have here, and I want to do it in a pretty fast manner where I will be going, for example, on one chapter of physics or chemistry or biochemistry or organic chemistry or whatever, and then try to just do them all together, and then like maybe switch up to a cars passage, and then switch up to back to the biology chapters, stuff like this. You get what I'm trying to do here. Just trying to be productive and trying to get shit done, basically. So where are we? Here we have the chapter 4 of biochemistry. However, we have not done chapter 3 yet. So it's a kind of gray zone for chapter 3. We did, however, go with chapter 2, and there's also all these biology chapters beforehand, all these things, and the types of questions that we can get from them. So let's make let's make goals. I always, when I worked out, I always used to do this. So let's write something here. Let's set goals here. So let's set goals as we have like 10, like one chapter overview. So for this, we have one chapter overview of biology. I mean, we don't have anything left for biology, so it's gonna be biochemistry. And then one chapter overview of organic chemistry. And then one chapter for general chemistry. And then we have something for even physics. And then we have psych-soc. We can also do one cars passage for the break, and then biology review, 10 flashcards. That's it. Let's make it 15. I want it to be the least resistant thing possible. Like 10 flashcards is just wait, and then we try to do more if we can in this session, right? So let's actually do start with biochemistry here, and let's go for the third chapter. So for the third chapter, we are going to cover the non-enzymatic functions of the proteins, and just like chapter 2 was about enzymes, now we're going to talk about, like what's happening with non-enzymatic proteins. So we have a bunch of cellular functions like structural proteins, molar proteins, binding proteins, cell adhesion molecules, CAM, or antibodies. We have biosignaling, we have protein analysis, and we have protein isolation. These are like kind of biotechnology, as they call it, right? So for cellular functions, we have structural proteins where they compose the cytoskeleton anchoring. So I'm going to go over it pretty fast. Anchoring proteins in much of the extracellular matrix. The most common structural proteins are collagen, elastin, keratin, actin, and tubulin. They are generally fibrous in nature. Motor proteins have one or more heads capable of force generation through a conformational change. They have catalytic activity, acting as ATPases to power movement. Common applications include muscle contraction, vesicle movement within cells, and cell motility. Examples include myosin, kinesin, and dynein. Dynein is for cilia and flagella, I believe. Like there is a primary ciliary disease where these dynein molecules are not functional, so the mucus cannot be extracted, cannot be taken away from our lungs, so it stays there, and that's why you can get infection from that. Infections such as pneumonia, I believe, or bronchitis. Then we have binding proteins. They bind a specific substrate either to sequester it in the body or hold its concentration at steady state. I think sequestering the substrate in the body means that you're holding it in the body. Then we have cell adhesion molecules, CAM, which allows cells to bind to other cells or surfaces. Cadherins, calcium-dependent glycoproteins that hold similar cells together. And integrins, they have two membrane spanning chains and permit cells to adhere to proteins in the extracellular matrix. And selectins, which allow cells to adhere to carbohydrates on the surfaces of other cells and are most commonly used in the immune system. So selectins, they're in the immune system. Keywords for selectins is carbohydrates and immune system. For integrins, they have two membrane spanning chains permitting cells to adhere to proteins in the ECM, extracellular matrix. And cadherins, calcium dependent glycoproteins that hold similar cells together. Then we have antibodies. We know them as Igs, used by the immune system to target a specific antigen, which may be a protein on the surface of a pathogen or a toxin. So the antigen is the protein on the pathogen. like bacteria, virus, or whatever. The variable region is responsible for antigen binding. Then we have biosignaling. So we have ion channels in action for biosignaling. This is like GPCR. These ion channels can be used for regulating ion flow into or out of a cell. We have ungated channels. They are always open. We have voltage gated channels like potassium channels, sodium channels. These open within a range of membrane potentials. And ligand gated channels, they open in the presence of a specific binding substance, usually a hormone or neurotransmitter. This is just like GPCR. Enzyme linked receptors participate in cell signaling through extracellular ligand binding, initiation of second messenger cascades. So the second messenger is the CAMP here, but the first messenger is actually epinephrine, interestingly. Then we have GPCR. It has a membrane-bound protein, like 7 domains, we know that, called the G protein, alpha, beta, and gamma subunits. So it's trimeric. The first messenger ligand initiates the second messenger and the cascade responds. Epinephrine is a ligand first messenger. See, at the end of the GPCR process, phosphodiesterase deactivates CAMP and GTP hydrolyzes back to GDP. So at the end of the GPCR process, when everything wants to go back to normal, this GDP that was once here comes back and replaces the GTP by hydrolyzing GTP. If you remember, with hydrolyzation, we can take a phosphate group out of this with phosphatase. So we have phosphodiesterase deactivates CAMP. And there is phosphodiesterase to deactivate CAMP. Then we have protein isolation techniques. So, for example, electrophoresis, it uses a gel matrix to observe the migration of proteins in responses to an electric field. We have native page. It maintains the protein shape, but results are difficult to compare because the mass or charge ratio differs for each protein. We have SDS page. So native, basically you don't do anything, you don't disrupt the protein, you just leave it as it is. So the shape is important. But the results are difficult to compare because the mass or charge ratio differs for each protein. Then SDS page denatures the proteins and masks the native charge so that comparison of size is more accurate, but functional protein cannot be recaptured from the gel. So functional protein cannot be recaptured from the gel. Then we have isoelectric point focusing, where you separate proteins by their isoelectric point. The protein migrates toward an electrode until it reaches a region of the gel where pH equals pi of the protein. So it's both charge at play here, and I think it's all charge. We don't know what it means here that the functional protein cannot be recaptured from the gel. We're gonna figure that out. Chromatography separates protein mixtures on the basis of their affinity. So affinity for stationary, phase or a mobile phase. Okay, so this affinity should be based on probably charges. Then here we have column chromatography, where it uses beads of a polar compound, stationary phase, with a non-polar solvent, mobile phase. So the solvent, the mobile phase, is non-polar, and the stationary phase is polar. We have to keep these in mind and come up with mnemonics for it, probably. Ion exchange chromatography uses a charged column and a variably salient element. Then we have size exclusion chromatography, which relies on porous beads. Larger molecules elute first because they are not trapped in the small pores. And affinity chromatography, which uses a bound receptor or ligand and an element with free ligand or receptor for the protein of interest. And for protein analysis of the structure, primarily determined through X-ray crystallography after the protein is isolated, although NMR can also be useful. So X-ray crystallography, after you isolate the protein, like we had here isoelectric focusing with one of these techniques. For the amino acid sequence, it's determined using the Edman degradation. We can research about these. For concentration, it's determined colorimetrically, either by UV spectroscopy or through a color change reaction. There might be actually questions from this. Bradford assay, BCA assay, and Lowry reagent assay each test for protein and have different advantages and disadvantages. The Bradford protein assay is most common. It uses a color change from brown green to blue. Beer-Lambert law, absorbance equals E extinction coefficient times concentration times L path length in centimeters. Okay, so we did, let's go with our, should be 5 to 10 minutes each. BC chapter 3 overview done. Now we have two options to continue with what we just read and do the flashcards for chapter 3, or just go on to the next one. In order to still bring this variation, we need to switch it up. So let's go with organic chemistry now. I hope it's at the end here. No. Let's see what we have before biology. So here we have organic chemistry. I did do the first 2 chapters. So for this, we will continue with the 3rd chapter. So the 3rd chapter of organic chemistry is about bonding. We have atomic orbitals and quantum numbers for the bonding. We have molecular orbitals. These are different a bit. We're talking about bonds, sigma bonds and pi bonds here. Then we have hybridization, and this should be a quick chapter. So for the atomic orbitals and quantum numbers, we have quantum numbers that describe the size, shape, orientation, and number of atomic orbitals in an element. So based on our periodic table, we are given an element, whatever, it could be iron, sodium, calcium. And based on that element, we can describe its size, shape, orientation, and number of atomic orbitals. So the size would be the n quantum number. It's called the principal number. Then we have L, the azimuthal. Was it azimuthal or azimuthal? Anyway, that's for the shape. For the ML is the magnetic quantum number, and MS is the spin number. So N labels for the energy, the electron energy level or shell number. So basically it's the size of the element. It's the size of the atomic orbitals actually in that element. And possible values could be 1, 2, 3. There's no 0 for N. Then, except for d orbitals, the shell number matches the row of the periodic table. So because for the d orbitals in the middle, it can be a 4s2 and then 3d3, something like that, but still the element is on the fourth row, not the third row. Then for the L, there is a 3D shape of that orbital. So the possible values would be like 0, 1, 2, etc. till n minus 1. So if you are on the 2s, then your azimuthal number is s. For s, it's 0 because n for the s is 1. And then 1 minus 1 is 0. For P it's 1. For D is 2. For F is 3. And for G is 4. It's just one less, right? Then for the magnetic number, and we'll see what the 3D shape of the orbital means, right? And for the magnetic number, we have orbital subtype, which goes from minus L to plus L integers. That's why for the L for S is 0. It's just one box that fits 2 electrons. For P it's 3, right? Because there's minus 1, 0, and plus 1. And for D is 5. It's minus 2, minus 1, 0, plus 1, and plus 2. And, oops, sorry. And for the spin, we have plus 1 and minus plus half and minus half. And the maximum electrons in terms of n, it's 2n to the 2 to the power of 2. So 2n 2. The maximum electrons in subshells, so in P or D or whatever, is 4l plus 2. So in, for example, D, the L is 2, right? We have it here too. So it's 4 times 2, which is 8 plus 2, it's 10. And for D, we do have 5 blocks, each fitting 2 electrons, which makes it 5 times 2, which is equals to 10. Cool. And in terms of N, again, we have like 4s, let's say. So S is 0. So sorry, S is 1. So it's 2 times 1 to the 2, it's 2. Then we have molecular orbitals. We have bonding orbitals and anti-bonding orbitals. So bonding orbitals are created by head-to-head or tail-to-tail overlap of atomic orbitals of the same sign. It requires less energy and it is more stable. The anti-bonding orbitals are created by head-to-head or tail-to-tail overlap of atomic orbitals of opposite signs. It increases energy and it is less stable. So the bonding orbitals are more stable, and they are head-to-head or tail-to-tail overlap of atomic orbitals of the same sign. So same sign means that these atomic orbitals are plus 1.5, for example. I think, but we'll see what this means. We have single bonds that are one sigma bond and containing 2 electrons. We have double bonds. There is 1 sigma and 1 pi. Pi bonds are created by sharing of electrons between 2 unhybridized p orbitals that align side by side. So they are unhybridized. That's why the electrons are shared. And triple bonds is 1 sigma and 2 pi bonds. Multiple bonds are less flexible than single bonds because rotation is not permitted in the presence of a pi bond. Multiple bonds are shorter and stronger than single bonds, although individual pi are weaker than sigma bonds. Yeah, due to less flexibility, because rotation is not permitted in the presence of pi bonds. Each pi bond, however, is just weaker than sigma bond. But in general, in total, the multiple bonds are shorter and stronger. And for hybridization, we have sp3, which is 25% S character and 75% P character. It has a tetrahedral geometry with 109.5 degrees bond angles. A tetrahedral would be like CH4. So you have 4 bonds with 109.5 degrees angles. You have sp2, which is 33% S character and 6%, 70% sp2 P character. It is a trigonal planar geometry with 120 degrees bond angles. So 120 degrees would be like this, and it's a trigonal planar. And we have sp, which is 50% S, 50% P character, and linear geometry with 180 degrees bond angles. H2O is not like that. H2O still has the angle, but it would be something like, I don't know, like CN2, I don't know. We have resonance describes the delocalization of electrons in molecules that have conjugated protein bonds, and conjugation occurs when single and multiple bonds alternate, creating a system of unhybridized p orbitals down the backbone of the molecule through which pi electrons can delocalize. Cool. I'm sorry, I'm getting pretty sleepy. I have to take a nap now. So I'll just put chapter 3 here. And we'll continue quickly with the rest. For now, peace.